DNA sequencing and mutations. Listed here is the DNA sequence we'll be utilizing in order to help us observe the works of transcription and translation, as well as various point mutations. Transcription and translation both occur anti-parallel to the template strand of DNA, so from the 5' to 3'. Transcription scans and codes DNA with its corresponding mRNA strand. Translation scans DNA for the allotted start codon and pairs each codon with its corresponding amino acids for protein synthesis. So listed above, I have our coding strand for DNA as well as our translated mRNA strand to help us compare. With missense mutations, these are changes of the amino acid which may alter protein function. So seen here with our AGC codon, our C is mutated, whereas it was initially AGG. So as that coded for the ARG amino acid, our mutated strand now codes for the SER amino acid. So with comparing it to our coding strand for DNA, we go from an AGG to an AGC. So because we have a G to C mutation or a purine to pyrimidine, that is also known as a transversion mutation. With a nonsense mutation, these are changes that code, there's a change of a codon that it becomes a stop codon. So as seen here with our UAG codon, which was originally AAG, this now converts from the lyse amino acid to a stop codon, therefore not allowing it to further read. So when comparing it to our coding strand for DNA, we go from an AAG to a TAG. So because we go from an A to a T, or a purine to pyrimidine, that is also known as a transversion mutation. So with our silent mutation, this pretty much does what the name implies. So this codes for the same amino acid. So as seen here with the GUU codon, which was originally GUC, GUC allowed for the VAL amino acid. UGUU also codes for the VAL amino acid. Now with the coding strand for DNA, we go from a GTC to a GTT. So our cysteine and thymine are mutated. And because we go from a C to T, this is a pyrimidine to pyrimidine, which is a transition mutation. Now read through is when a stop codon is changed from a codon that codes from amino acid, allowing for the sequence to be read through what used to be a stop codon. So as seen here with the UAC, which used to be our stop UGA, it now codes for the TYR amino acid. So we go from a TAA in our coding strand to a TAC. And because it is from A to C, is a transversion mutation.